Okay, so today this presentation is going to be going through our mid-class material for February 9th. So this is our snow day. You can look at the class slides, but even better, go through this brief narration of, it's not actually all that brief, narration of the slides that I'm putting together here. So we're going to start with color and how we label color using an RGB system, and then talk about color vision, which is really, really super important. And I want to make a note again on homework that if you don't know the answer of something that is being asked in the homework, do ask about it. This is the reason to have a homework question. If you can't do it on the homework, then when it rolls around again, when you're trying to use it in something else, in a problem or a project, or when it comes up on an exam, you also won't be able to do it. So let's talk about that. The particular one in this homework was the frequency calculation. So we're going to start by having you go to a video called There Is No Pink Light. And this is a 1-minute physics video. So take your time, flip over to that video. Hopefully, if you copy and paste this entire link, you can find it. But it is on YouTube called There Is No Pink Light. And what I'd like you to think about is the relationship between the spectrum that we already looked at and then this discussion of what is pink light. Okay, so going on to color identification, what we use in our system, color system, is 3 colors, red, green, and blue, to identify every single color that we can see. And yes, I'm going to go on with red, green, and blue through this entire class. Soon we're going to talk about why the primary colors are sometimes red, blue, and yellow. But that's in painting, that's in pigments. So today, red, green, and blue are our primary colors. So again, another video on color mixing and the pink and magenta question. This video is a very nice demonstration of what happens, basically illustrating the little symbol that's in our top right corner here of our slides, illustrating what happens when you overlap certain colors and what you get. So take the time to watch this video also. If you have any trouble with the links, do let me know. They're not links for you, so if you have any trouble accessing them. Okay, so after that video, now that you've seen it, let's just review. When we combine only red and green, what do we get? Think about it for a moment. Pause here. And when we combine red and green, we get yellow. So there we've got the overlapping red and green, we get yellow. Okay, when we mix green and blue, what do we get? And the answer is we get this combination of green and blue, which we call cyan. And cyan is now the hue that we see when we mix green and blue. And when we mix red and blue, what do we get? The answer here is magenta. So magenta is the hue. Hue is the language for the color. There are a couple different ways of identifying color. And you've seen the reading, which uses the color triangle. It comes out of a slightly off, differently shaped triangle, but what we've seen in our book is the triangle as shown on the left, where the bottom axis is all red and the left side axis is all amounts of green. And when we look at just amounts of red and amounts of green, we can describe every single color. Also, we can use this color triangle to find complementary colors. So for example, if we want to find the complementary color of yellow, we start with by making a dot at yellow. We draw a line through white, which is in the center of the triangle, and we go the same distance across, and we get something that's kind of a blue. Spectral blue is what that means, SB. And it's not all the way to the end. This is really the complementary color of that yellow. So what I'd like you to do on the next slide is look at one color, pick one of them for about 15 seconds, and pause it, and then go to the white screen. Go on to the white screen slide. What you should see is a complementary color, and then check it. See if you can tell which complementary color matches the original colors. So this is your initial slide. I'm going to go on. You can pause here for as long as you like. And then you can use the white slide. So how do we use this red, green, and blue system to describe colors? Colors are basically either all red, all green, all blue, or have a hue that's somewhere in between red, green, and blue. And those hues are what we called cyan, magenta, and yellow. So what we do to label them is we pick a relative power of red, green, and blue. So to remind you what a percent is, if we see 0.3, that means 30%. So if we had a number, so this color down here is RGB is 0, 0.3, and 0. Let's think about that. So red, green, blue. The 0 is green. Sorry, the 0 is red. The 0.3 is 30% green, and the other 0 is blue, which means it's definitely going to look green. And in fact, we call that saturated green. So that third color, the color that is still present, and the other two components are 0, is our saturated color. Okay, so we're going to go through an example, and this is what the worksheet that you're going to do is based on. Let's say we have RGB given as 0.3, 0.7, and 0.65. What color is that? So the way we work that out is we break our color into a saturated component and a gray component. So what is gray? Well, gray is anything that has equal amounts of red, green, and blue. So we take the smallest percentage, in this case the 0.3 is the smallest number there. And we're going to separate out, we're going to subtract out 0.3 from all three of those numbers. So I'm going to take out, here this is my gray component, I'm going to take out 0.3, 0.3, 0.3. And what I'm left with is 0, so 0.3 minus 0.3 is 0, 0.7 minus 0.3 is 0.4, and 0.65 minus 0.3 is 0.35. Okay, so our color is now broken into a gray, which tells us just more about the intensity of the color, and a green and blue component. So if there's green and blue, it must be cyan. And so that's just one example. It's your worksheet and your homework asks you to try other examples that are like this. So do come and talk to me about working out these examples if this doesn't make sense. The way to find the saturation level is to break out your color into a color that has at least one component that's 0. And that's called your saturated power, your saturated color. So in our case, our saturated component is 0.4 and 0.35. We're going to add those two together, 0.4 plus 0.35. And we're going to divide it by the total original power, 0.3 plus 0.7 plus 0.65. And what we get is what we call the saturation level, or 0.45, of the color. So what we're asking ourselves is how much of this color is really going to show up in how we see it. How green is it? How cyan is it? The higher the saturation level, the more color you'll see, the more we'll think of it as a brighter color. This relates to the worksheet 4.1, that you're going to do after watching this video. And one more video. This is a great video of Bill Nye the Science Guy, talking about color, light and color. And think about or notice every time that he mentions additive color mixing or anything that has to do with additive color mixing and subtractive color mixing. So to back up, what we've been talking about is additive color mixing. What we'll be talking about when we do paints is subtractive color mixing. And now we're going to make a shift to the next half of our discussion today. This is a really important part of the discussion. This is what I'm really sorry that we're missing in class, so stick with it. The slides here are almost all thanks to Bruno Richard, who is at Concordia University in Montreal, who visited my class last year. The spherical cow is simply, or the cow in a sphere is simply a recognition of spherical cows. And you'll notice that he's Canadian and he spells color with a U. All right, so just to get you started thinking about color, you can read the numbers. It's kind of fun. And if you can't read the numbers, you probably already realize that there are different colors. Just like I can't see depth and someone shows me a 3D image, it doesn't make any sense to me. If you can't see colors, the numbers may not stand out. We're just all different in the way we see. All right, so color perception. The question would be something like, why do strawberries look red? And the answer is that it has to do with the physical properties of light. And a better question might be, why do we care? You know, one answer would be that if we didn't have color, this photograph is kind of dull. But when we add color, we can see those strawberries in there. And it's a lot more interesting. I don't know that those are strawberries. I think they're raspberries. Although they might be. Okay, moving on. Let's remind ourselves of the visible spectrum, which we should feel very fluent with by now, going from the blues to the reds within our entire electromagnetic spectrum. And thinking about color goes back to Isaac Newton, actually. We think of Newton for Newton's laws in physics, or you might. And this is a fun quote. "The rays, to speak properly, are not colored. In them, there is nothing else than a certain power and disposition to stir up a sensation of this or that color. So colors in the object are nothing but a disposition to reflect this or that sort of rays more copiously than the rest." And that is exactly what we're talking about with color vision. And this is we've seen this before. If you stare at the black dot for a while and then shift to the next screen, you will see the complementary color appear in your vision, and you'll see a castle with a blue sky. But we'll just move on to talk more about strawberries. So what's going on is all of these different rays of light shine on a strawberry. In other words, sunlight, which is made of a lot of different colors. This would be the spectrum of sunlight, shines on the strawberries. And what's actually reflected off is just the orange and reds, because the strawberries subtract out all the other colors. Then it goes to our brain, first into our eyes, and then to our brain, and what comes out is this puddle of red. That's how we interpret that information. There are different ways of thinking about color perception. This is an old drawing, an ancient drawing of sensory information and how it was thought about. If we look on the left here, there are a couple parts of color perception. One is psychophysics, which is the quantitative relation between the physical and psychological. So the psycho being the actual perception that we have, and the physics being the physical nature, like the light, the wavelength, the numbers that I've been talking about. That makes it quantitative, the connection between perception and the physical nature of the light. The physiology is about identifying the mechanisms that are responsible for this perception, this vision that we're talking about. So here's an interesting thing about the visual field. When you look out, there's the left side of your visual field and the right side. So that would mean the people sitting in front of you in a row, there'd be some on the left, some on the right, say, and those on the left are the left side of your vision. Okay, so the rays of light from both the left and the right side travel into both eyes. But let's see what happens with the left rays, the ones that are in red. They go into your retina and they strike the far right side of your retina. From that right side of the retina, those rays in red travel through the optic nerve, and some go to the right hemisphere, and some go into the through the chiasms, optic chiasm, and some go more into the left. So you actually get some input into both sides of your brain from the left side, but primarily goes to the right side. And similarly, the input from the right side of your vision goes primarily, the blue lines, to the left side. So a picture of the fovea. You've seen similar pictures that are actual sort of diagrams of this chart, and we'll see, or this picture, we'll see more. So if we take a slice of the fovea, we see rods and cones all the way across the fovea, and that's what it's made of. It's just these actual cells, these things in our retina called rods and cones and other stuff. If you've ever had any injury to your eye or any reason to look at your retina, you may be too young to have experienced that, you'll get pictures of your retina. This thing over here is the optic nerve. And we could check back to the previous slide and figure out whether this was your right eye and your left eye. We'll come back to that another time. Think about it. Is this your right eye or your left eye? All right, so getting into a little more details of what's on the retina. We have a lot of parts of the retina. The fovea is the part of the retina that's really important for vision, for precise vision, for making out fine lines and details, and also for color. We're going to see that in a moment. And you can look at this nice picture also in your reading about how the different cells are broken down in the retina. So this layer is what we're going to focus on next, where we have photoreceptors, rods and cones, and then this is describing the inner and the outer segments of the rods and cones. So on to rods and cones. The rod looks like a rod, and the cone looks like a cone. The cones are a little bit shorter, the rods are long and narrow. They both have an outer segment and an inner segment, and then a nucleus and synaptic terminal. The rods, you can see this picture. They look very similar to the diagram, the schematic on the left. The good things to know, well, the important things to know, rods are really for night vision, and they turn on with very low illumination levels. They have no color. You only see gray. Have you noticed, for example, that when it's dark, you don't really see color? And that's not just because it's dark, it's because your cones aren't on, but your rods are. So you're seeing just grays, levels of light, but not color. The cones, on the other hand, are really specialized for daylight, and they respond very well in high illumination. So they're responsible for our color vision and our detailed visual acuity. They're all centered in right in the center, right in the fovea. This describes where the rods and cones are. You've seen this chart a couple times now in a couple of textbooks, so you must be thinking, okay, this must be telling us some useful information. The red line in the chart or in the plot, is our rods. So there are like no rods right at the center of your vision, where you're dealing more with color and light, very light things, and also visual acuity. And there are many, many rods out more on our peripheral. In contrast, there are a lot of cones right at the center of our vision at the fovea, and no cones, or very, very few cones, out at the edges of our vision. The optic disc is always drawn here, and it just blocks out everything. There are no retinal cells there in where your optic disc is. So we're going to talk about color vision. This is kind of cute. Basically, normal color vision, we can detect about 2 million different colors. Pretty exciting. And then if there's really, really good illumination, you can see even more variation in color. So how do we do this? The questions, it's fascinating. We're still figuring it out. We have graded responses, which means greater or lower responses at different, with different levels of light, different colors. But maybe they just scream. Maybe they're sending Morse code. It's kind of it's fascinating to see how this all works. We are not going to go in depth into the theory right now, but there are two theories: trichromatic theory and opponent process theory. What we're going to talk about, though, is trichromatic vision. So this chart you'll see about 3 times in the next 2 minutes. And it talks about our 3 different types of cones and our rods versus wavelengths. So by now we're familiar with wavelength. Short wavelength over on the left, more blue light, blue colors. Long wavelength over on the right, more red light and red colors. And 3 types of cones. The S cones are S for short wavelength. The M cones for middle wavelength, and the L cones for long wavelength. Notice that the rods here are just picking up light, but primarily respond to wavelengths centered around 500 nanometers. Okay, so if you have not seen the radio or heard the Radiolab episode on color, it's excellent, and it talks about this. We are not hexadecachromats, but the mantis shrimp is. So it has 16 different types of color receptors where we only have 3, which means this where we see a rainbow, the mantis shrimp would see a thermonuclear bomb of light and beauty. Very cool to think about, and we don't even know how to color all of these cones. We don't know what color to give them. They don't have color for us. So the mantis shrimp is the winner in our world that we know so far with being able to see color. Okay, so I did not choose the photo, but it does show very clearly what happens with different types of color blindness. So it's an interesting progression. Rod monochromats are really colorblind. They only have rods, no functioning cones. So that's what it looks like when you're a rod monochromat. Someone who has protanopia has a malfunctioning L cone, which means they have poor red and green deficiency, or poor red and green differentiation. So that's what it looks like if you're a protanope. If you're a deuteranope, your second cone doesn't work very well. Similar malfunction, you get a similar final effect in the color blindness. And tritanopia, if you're a tritanope, you have poor blue and yellow differentiation, and your colors would look more in the reds and blues. Right, so again, seeing this chart of the S cones, the M cones, and the L cones, and the rods in between, this is our take-home message for rods and cones. To give you a test of what your rods and cones are doing, if you look for a while at the white dot, and then hold it here for about 10-15 seconds, and then go on to the next slide or next image, and you will see the American flag in all of its color glory. So how do we see colors that aren't part of our cones? And this is the thing that we talked about with magenta early, and it has to do with just adding colors. We actually pick up different amounts of red, green, and blue. We combine them all, and we eventually see something that looks yellow. So we can write yellow as a combination of red, green, and blue, whether we're adding them or taking the difference, and end up with something called yellow. And that gives us all of the hues that are in this picture, and leads us to have color vision. To remind you about office hours, you can come back here to this slide, and also to remind us what we're going to be doing on Thursday is meeting up in the library, and you'll get more information about that. Check the Blackboard site, check our Blackboard announcements page for the most up-to-date information on readings. The readings for Thursday is just from Goldstein. It's fairly short. And that's all.